Good evening, one and all. On behalf of SSC LMP, I, Dr. Santosh, would like to welcome you all to the concluding session on gene therapy. Yesterday, it was a very informative session, very nice and lucid presentation of a very complicated topic. So today, we will continue with the remaining part of gene therapy. Hope this academic endeavor will enrich us further. I would request all the participants to post their questions in the chat box. We will be able to answer maximum 4 to 5 questions due to time constraints, and I would request the participant to forward the other questions to the official SSC-LMP email ID that I will put in the chat box. Now, without much more ado, I would like to invite Dr. Anindya Dasgupta, Professor and Head, Department of Biochemistry, Medical College, a wonderful academician, to continue his session on gene therapy. Welcome, sir, and over to you.
Okay, Dr. Shantasmita, thank you so much. So yesterday's, I think that yesterday's session, it was the starting session, and today's session is the concluding session. But after today's session, it will be clear to all of us that this concluding session is the starting session, in fact, for the gene therapy, in fact. That means how we understand the gene therapy. This will be the starting of our knowledge. So with this brief introduction, I start my class. I think that everybody is able to see the slides. Okay?
Yes, sir.
Okay.
Yes, sir.
So yesterday we stopped at this point that the strategies for RNA repair, and we have discussed this trans-splicing using spliceosomes, trans-splicing using ribozymes, and we have also discussed how this spliceosome mediated RNA transcript technique can cure some of the RNA mutations. Now today we will start at a very interesting thing, that is how the double-stranded RNA molecules, because they are foreign RNAs to us, because we know that eukaryotes generally they have the single-stranded RNA molecules. So when we have these double-stranded RNA molecules in our body, they are going to do something new, and that was discovered by these two scientists, that is Craig and Mello, that who in fact Craig Mello and Andrew Fire, in fact, these two scientists, they accidentally discovered, just like many other scientific inventions, this silencing of the gene by introducing a double-stranded RNA, and they named this as simply small interfering RNA or siRNAs. Now, what happens that when the double-stranded RNAs or these siRNAs they enter in our body, generally they are found in the bacteria to defend themselves against the viral attacks, and in eukaryotes, in the present days, they have been discovered also in the eukaryotes. So, as double-stranded siRNAs, they are cleaved by the enzyme, that is an RNA cleaving endonuclease enzymes, particularly known as DISAR, and into this long chain, that is siRNA, is cleaved into a single-stranded RNA molecule, and this single-stranded RNA molecule now combines with a protein complex that is known as RNA-induced silencing complex, with some Argonaute family of proteins which also belong to this RNA endonuclease or RNase enzymes. The job of this RISC is to guide this Argonaute complex and this guide strand of this cleaved single small interfering RNA molecule to the target mRNA eukaryotic molecule and cleave it down. So this can very well cleave any defective mRNA bearing the disease genes or defective genes. Now, there is another thing that is known as microRNA. Now, there is a very confusing grounds. There are some confusing grounds. The students, particularly the postgraduate students, when they come to this field of this double-stranded mRNA, they feel a great confusion between this microRNA and siRNAs. So here, just I have mentioned the salient differences between them, as both of them can modulate the mRNAs, but in a little bit different ways. Now, mRNA, that is the microRNA gene, they are naturally coded by your RNA polymerase II enzymes. And then they are inside the nucleus, in contrast to the small interfering RNA molecules, which are found in the cytoplasm generally. So inside the nucleus, these microRNA molecules are processed by these enzymes. They are excreted or transported inside the cytoplasm. You find the similarity with the siRNA is that they too bind with this RNA-induced silencing complex or RISC and this Argonaut family, superfamily of endonuclease proteins, and they form this complex. But the big difference is there that they are not bound to their target mRNA completely because they have got some mismatch here. In contrast to the complete binding with the small interfering RNAs with the target. The result is that the small interfering RNA can bind only with a particular target because it is very much complementary to the target. But these microRNAs, as they do not bind completely to their complementary RNAs due to this U loop, it can bind with a multiple targets or multiple mRNA targets. So microRNAs can regulate gene transcription and have found in the place of gene therapy field in a more versatile way than these small interfering RNAs. So this is the thing for both. Passenger strand means the sense strand for both, leaving the antisense strand intact, and that antisense strand is going to bind with the sense mRNA strand to silence it or to cleave it. Okay. Now, another difference between them is that the small interfering RNA, as you can see, it only cleaves, it only destroys the target mRNA. But the microRNA, as they do not bind completely with their target mRNAs, they can suppress the protein translation from the mRNA, they can modify the mRNA, and they can also cleave the mRNA. So their mechanism of action is also versatile. Now, if we want to put them in a nutshell, then these are the differences between them. I have already mentioned them. The major points of interesting is that the target of siRNA is only one mRNA, and the target of microRNAs are multiples. They can be more than 100 at the same time, so they are much more versatile. A huge array of human diseases, they can be treated by this double-stranded RNAs. That means both the siRNAs and the microRNAs, together we call them both as RNA interference, Now, what are the challenges? Because if we don't understand the challenges of the gene therapy, we cannot move forward more. Now, the introduced small RNAs tend to be degraded by nucleases in blood within minutes. It is true for every RNA molecules. So generally, the more popular method of transmitting them into the cell are the local administrations, like intranasal and intravitreal administrations. The second one is that you can also modify their degradation, or you can restrict their degradation by modifying, by adding something, say for example 2O methylation of the antisense strand. The second challenge is that Non-specificity of delivery to the target tissues. It is the common challenge of every aspect of the gene therapy that we have seen till now. So what is the way out? You can package them in the lipid lysosomes. If, for example, for the vascular endothelial growth factor mRNA, if you target against the VEGF mRNA, this is very successful mode of transmission. Package them into lipid lysosomes and use nanoparticles. You tag these liposomes and nanoparticles with specific antibodies, and then you can see that they will reach their target cells. So this is the liposomes and use of nanoparticles. The third challenge is that they may introduce interferon response if not designed properly. That means if the length of the introduced siRNA or the microRNAs, generally they should be within your 21 to 25 nucleotide of length. But if by accident you design them such a way that they become more than 30 nucleotides long in double strands, that means when you introduce them for the first time as double strands, and if that double strand is more than 30 nucleotide long, they can induce severe gamma interferon response and immunological assault. So you have to keep them limited within this 30 nucleotide. Now this is the thing that I was talking in the previous slide, just a schematic representation that liposome mediated transfer, you just pack them within the lipid molecules or liposome molecules and then inject them with their targets with the specific antibodies, and they will reach their target cells. In the same way, you just tag the nanoparticles with the antibodies specific to the particular cell, pack the siRNA or miRNA, whatever you want to use, into that nanoparticle and deliver it. Then it will reach to the target cells, and by receptor mediated endocytosis, as you can see there, it will enter into the target cell and do its function. So these are the two common methods to increase the targeted delivery of the double-stranded RNAs, or to increase the benefit of your siRNAs and microRNAs. Now, till now we were discussing about the double-stranded RNAs because they are foreign to us, and that's why they can very effectively be used to treat our genetic diseases. Now we have also some single-stranded oligonucleotides which can do these jobs, but not as efficiently as these siRNAs or miRNAs. But still, then we have to know them a little bit. So I've just discussed about a little bit of them. The single-stranded oligonucleotides which do these jobs, generally they are called as antisense oligonucleotides. So what they do? They can render the target gene or the target mRNA more susceptible to RNase H. They can introduce or induce alternative splicing sites using oligonucleotides against the splicing sites. And they can also block its expression. Oh, well, I think that we can go to the slides, pictorial slides directly. See, this is an ASO, that is antisense oligonucleotide gapmer. Gapmer means these oligonucleotides has got a central region which can make the target complementary RNA molecule, that is eukaryotic mRNA molecule, much more susceptible to the enzyme that is RNase H. So this RNase H will be cleaving the target mRNA. As this particular gap is filled up by a particular sequence which makes the target mRNA more susceptible to this RNase enzyme, this molecule, a single-stranded oligonucleotide molecule, is very, very common and is known as ASO gapmer to treat targeted gene destruction, defective gene destruction in the RNA molecules. The second one is a particular oligonucleotide that can suppress the translation, and so they are named as translation suppressing oligonucleotides. What they do? They just bind at the vicinity, close proximity of the start codon, that is AUG, on the mRNA, and block the ribosomal subunits to act on this start codon. And in this way, they block the protein synthesis or the translation mechanism. The third type of antisense oligonucleotides, they can act as a guide sequence to mislead the target mRNA to the ribonuclease P that is very well found within our cells, and ribonuclease P will be very happy to get the mRNA trapped within it, and this will degrade the mRNA. Okay. So this job of this external guide sequence, antisense oligonucleotide, is to mislead, is to bind the target mRNA and mislead it to the RNase enzyme that will destroy it. So these are the different types of RNA repairs we studied, starting from the spliceosome mediated RNA repair, ribozyme mediated RNA repair, double-stranded like your microRNAs and small interfering RNAs mediated RNA repair, and the single-stranded antisense oligonucleotide mediated RNA repair. Overall, these all types of RNA repairs are used widely in the treatment of these disorders. You can see them widely used in the treatment of these types of disorders. So they are very useful. In fact, they are much more versatile than the DNA-based repair systems. So after learning these things, now the thing is that who will bail the cat? That means how these modified genes or the therapeutic genes will be delivered inside the nucleus, inside the RNA, by which vector, by which vehicle, etc., etc. So now let us go there. Okay. So tools for transfer of this introducing different genes into the cells for gene therapy may be direct transfer, or they may be indirect using several vectors. So direct transfer means either you inject directly them into the nucleus, that is by the method of intranuclear injection, transfer the nucleus with some chemicals like calcium phosphate, or use electricity to simply cause electroporation. On the other hand, the vector-mediated transfer, they can use several viruses like adeno associated, herpes simplex, and some plasmids. Now the thing is that since the advent of recombinant DNA technologies, we have got several these pre-owned genetic combinations ready for use. The only problem is to deliver them to the cells where they are needed. So we have got only two choices. What are they? Either you need the target cell out of the body to be modified and then reintroduced, or you need to introduce the genes into the cells using some effective vectors, that is in vivo. So using these two methods, whatever method you use, you have to enable the modified therapeutic gene that you have modified in the laboratory outside the cell. You have to enable that modified therapeutic gene to enter into the cell, whether it is ex vivo or it is in vivo. So how we can do this? Okay. But before that, let us know the basics of this ex vivo and in vivo gene transfer. This is what we will define it later. What we are doing here, we are making copies of therapeutic genes. Then we have inserted the genes into the viral DNA, that is the vector DNA. And then we have cultured cells which have been infected with the genetically altered virus vectors. And what is the number 2 sequence here? Target cells, they have been removed from the patients. That means we have removed the cells from the patient itself. You have taken, suppose you have taken out the lung cells from the human being, we have grown these defective lung cells which are more prone to develop lung cancer, say for example, or some p53 mutation into a culture plate, and into that defective cell you are introducing the therapeutic gene. And then what you are doing, you are reintroducing the therapeutic gene modified patient's own sample into the patient's own body. And inside the body, these genetically altered cells, they will definitely produce the desired protein encoded by the therapeutic RNA or the therapeutic DNA. So what type of this thing? Therapy is known as ex vivo gene therapy. That means we are modifying the cells ex vivo. That means outside the body. And then we are introducing it into the human body. In contrast, the in vivo, or the opposite thing, is that let the cells not to be taken outside the human body, let them remain within the human body. Whatever modification you need to do, you do it within the human body. So that is known as in vivo gene transfer. Okay. So basic thing of any viral vectors is that either it is a viral vector or non-viral vector, a therapeutic gene must consist of these three things. One is the transgene, that means the modified therapeutic gene. Another is the promoter region for enhancing the transcription and translation of that gene. And then it is the terminator region that stops the protein synthesis. Together, we call them three as the open reading frame or the effective transgene expression cassette. Transgene expression cassette. So in the therapeutic gene cassette or the transgene cassette, we can use the direct methods. That is, as I told you, by electroporation, you can enable the cells you can enable the therapeutic gene cassettes directly into the cell by just what thing? Give an electric pulse. These green things are the therapeutic or the modified plasmids or modified genes. After a brief electric pulse, the small pores or micropores will be there in the membrane of the target cell, and the therapeutic genes, they will get their entry very happily within the cells. But you do not know what percentage of the cells get entry. It may be 5%, maybe 1%. So you may have to repeat it. Second one is the chemical method, other than the electroporation method, that is used by using the calcium phosphate. Calcium is a very common ion that can mobilize the plasma membrane. You know, its calcium means motility. So wherever it is present within the muscles or within the plasma membrane, it will induce motility. So here also what it does, you are using tagging calcium phosphate molecules with the therapeutic genes, and you find that they are inducing mobility in the plasma membrane, and ultimately they get entry into the cell. We call this process as DNA or RNA transfection. Okay. The third one is, that is the direct transfer without use of any vector, is the gene gun or gene gun barrel, where you can in fact spray the DNA molecules or the recombinant DNA molecules at a very high speed to the target cells. And this is particularly useful for the plant cell molecules where the coating of the plant cells are very tough. But in human beings or the other animal kingdom cells, this thing is generally not needed. But the most modern theory or the technique for introducing the genes directly into the body or into the cells, they are known as hydrodynamic gene transfer, that has been discovered not many years ago, most probably in 2013. So this is relatively a very fresh technique. What is done here, that in the hydrodynamic gene transfer, you inject a large amount of modified genes, large amount of modified genes you can inject into a small capillary, particularly that type of capillary that surrounds the parenchymal cells. Now, due to increased pressure of the injected large volume of the DNA or the RNA or therapeutic gene, the pressure inside the capillary increases. And as the parenchymal cells, mostly they have the fenestrated capillaries. So the genes enter through the fenestrated capillaries into the parenchymal cells. You can see the parenchymal cells have swollen up here. Okay. Parenchymal hepatic cells, they have swollen up here. That means they are taking the fluid from the capillary endothelium. And if you tag these genetic elements with green color, green fluorescent color, after some time you will find that the cells have become, cells have gained the same color as the tagged external genetic elements. That means the cells have taken the genes successfully. Now, this hydrodynamic gene transfer model is particularly useful for treating or having gene therapy for the hepatocytes. The precondition is that you have to have generally a fenestrated capillary endothelium so that the liquid containing the DNA can directly go from the capillary endothelium to the parenchymal cells. Now, what are the different challenges? Challenges mean that it cannot happen that I have introduced the DNA and it will very happily go through the, or I have introduced the therapeutic gene and it will very merrily go into the body. Nobody will be attacking the genes. They will happily travel through the bloodstreams and they will directly enter into the nucleus. This thing never happens. They have to face several challenges. What are they? First one is transfer across the hydrophobic membrane. Because negatively charged DNA molecules, they tend naturally to resist the transfer across the hydrophobic biomembrane. You know, negative means they are hydrophilic. So how the hydrophilic molecules can be allowed across the hydrophobic membranes? And second one is the chance of definitely nucleus digestion in the plasma. So how to get rid of them? First, you condense simply the plasmid vector, if it is plasmid vector, with some, this is particularly suitable for the plasmid, so I have mentioned this as a plasmid vector, with cationic lipids, okay, converting them into lipoplexes or lipid droplets or lipid aggregates, or you can convert them into liposomes. And the second one is convert them into a nanoparticle covered media, which is covered by cationic polymers. So this nanoparticle mediated transfer and this liposome or lipoplex mediated transfer can very easily carry the artificial gene across the biomembrane. But even when they pass through the biomembrane, they face some immunological risks. What are they? First of all, the toll-like receptors, which are present in our body from the very birth of us, from the very beginning, from the birth of us, which do not depend on any external attacking antigen to be stimulated. So because they belong to the innate immunity, and they just identify some patterns, and by identifying these patterns, they have definitely identified that this foreign molecule or engineered molecule that has been introduced into my body is foreign to me. It is not bearing my recognized patterns. So these toll-like receptors, they will attack the endoplasmic vesicles containing the therapeutic genes and destroy them. But we have also discovered some ways to circumvent. The most important is of them are you convert these things into a disguised form of artificial DNA, that is in form of mini circle DNA. Mini circles DNA cannot be recognized by the toll-like receptors. They are just like those aeroplanes which can defy the detecting signals, which can defy the radars. So they just bypass the radar system of your toll-like receptors, and they will not be attacked by the immunological asset. That's mini-circular DNA, and this mini-circular DNA, they are synthesized by a particular technique that is known as FLC. You freeze the DNA, you lyophilize the DNA, and then again cyclize them. Convert them into cycles by cyclization. So converting this artificial DNAs into mini-circular DNA, you can avoid the nuclear atom. The major problem occurs of introduced DNA. Even if it enters into the cell, then it remains, particularly if it is the DNA viral vector, then it remains as an episomal DNA because it is not integrated into the nuclear DNA. And being an episomal DNA, what happens, that nuclear DNA goes on healthily just dividing it, it will go on increasing its number, but not the episomal DNA. They will not enjoy the same cell cycle facilities because cell cycle is different for the nuclear DNA. So they will gradually be reduced in number. But we have also got the remedy for that. What we have done, we have put out a fine segment of the nuclear DNA and tagged our artificial gene that has been introduced into the nucleus with this. So this scaffold matrix, it contains a small part of eukaryotic DNA sequence which has been attached to the vector. And so now the vector doesn't remain far away from the nucleus, but goes very close to the nucleus as it contains the eukaryotic DNA segment as a scaffold matrix attachment factor. And it remains tagged at the outside wall of the nucleus very closely. And that's why whenever the nuclear DNA divides, it also divides. So this is a very good engineered DNA by using these scaffold matrix attachment regions and attaching them into the foreign DNAs, therapeutic genes, so that they can enjoy the same number of cell cycles as the nuclear DNA enjoys. So after going through these things, that means these non-viral vectors that we are still now studying. We have studied how to introduce the non-viral vectors by different techniques like electroporation, like your chemical induction, like by hydrodynamic gene transfer, etc., etc. Are there any advantages of them? The simple advantage is that as they do not contain any viral part, their direct transfers, the chance of immunogenicity is very less. And as there is no limitations for introduction into the viral genome, large amounts of DNA can be directly inserted. And as you do not need any virus engineering as viral vectors, cost of production is also low. But the major difficulty is that they are not allowed to enter into the cell with very simplicity, because a great difficulty always occurs to enable them to enter into the cell, and very low percentage of them can enter into the cell only. So the net result is that although recent techniques, they have increasing the transgene expression using DNA vectors, this method is still far away in competency for gene transfer in comparison to the viral vectors. Just because we want efficiency. Efficiency means we want that the amount of gene that is transferred should match with the amount we wanted for. So we go for the viral vectors, like your the viral vectors can be both type of DNA or DNA and RNA viruses, as you know, as they are expert in infecting the mammalian cells, and they can also use specific receptors via endocytosis for their entry, and most importantly, they are quite expert in integrating into the genome of the host, being retroviruses. So having these advantages, uh, generally the viral vectors for gene transfers are engineered. This is a wild type of viral genome. You just take out the viral genes and insert your target therapeutic genes and form the transgenic acids. So once the transgenic acid is ready, you can introduce it into the virus, and the viruses commonly used are the gamma retrovirus, lentivirus, that is also a type of retrovirus, murine leukemia virus, that is also a type of retrovirus, and now these three things, that is adenovirus, adeno-associated virus, and herpes simplex virus, they are the DNA viruses. The first two that we are going to discuss now is the gamma retrovirus and lentivirus. Now, gamma retrovirus and lentivirus both are retrovirus, there is no doubt. But the one thing is that gamma retrovirus can replicate or can only infect the actively replicating cells, while lentiviruses can infect both replicating and non-replicating cells. So definitely it is very easy to understand that lentiviral vectors will have more wide acceptance. Challenges are there always, that you have to make them specific to the particular target cells, and you can do that very easily simply by adding the receptor glycoprotein molecules or an antibody that is specific to an antigen of a particular tumor cell with the viral capsid molecules. So definitely the virus will be guided by its cell glycoprotein or the antibody receptor on it to the particular cell type containing that particular antigen. But the major danger is there that retroviruses are double-edged sword, because they can insert themselves into the host genome. So the basic problem is that if they do not insert themselves into the right position and they insert instead in wrong positions into a healthy gene, they inactivate or they convert a healthy gene into a mutant gene, then this is known as insertional mutagenesis. Insertional mutagenesis. Insertional means insertion mutagenesis due to insertion of a retroviral genome, that we call as proviral DNA or proviral mRNA. The simple proto-oncogene can be converted into a very active oncogene. So you have to get rid of that also or minimize that. So the scientists have also done this. They have converted or equipped the retroviral vectors with a self-inactivating vector design, SIN. What they do? It has been found, if you have read about the insertion of the proviral DNA from the retroviral molecule or proviral RNA from the retroviral molecule, the long terminal repeats of the virus, that means if this is the virus molecule, it has got several long terminal repeats at its 5-terminal and 3-terminal end. Now these long terminal repeats are known in short as LTRs. And these long terminal repeats are responsible for enabling the viruses to be integrated in wrong places particularly, or converting the proto-oncogenes into oncogenes. So what the scientists they have done, they have removed the promoters and enhancer regions in the long terminal repeats. So if the long terminal repeats are inserted now, there will be no promoter and no enhancer here. And as there are no promoter here now, so even if the LTR is introduced, it cannot activate any oncogene or any proto-oncogene into an active oncogene. Okay. This is another novel way by introduction the zinc finger nucleases into the RNA virus molecules, particularly the retroviral molecules. We are well acquainted with the zinc finger model. They are nothing but zinc finger models are the DNA binding proteins, you know. Now, generally what happens, these DNA binding proteins when they bind the target nucleic acid molecule, they provide cleavages as well as they provide some off-target cleavages. That means if this is the target, then target cleavage along with some off-target cleavage like this area will be also cleaved out. So whenever this will be cleaved out into two parts, there is chance of insertion in mutagenesis here. So you can block them also by modifying these specific zinc finger nucleases that are the particular nucleases which carry out this off-target cleavages. What do? You just add some extra zinc fingers here. That means these are the extra zinc fingers that have been added here. Here also. And after modification of this thing, that means creating some extra fingers, these zinc finger nucleases will no more cleave at any off-target regions of the DNA. So if there is no off-target cleavage like this here, there is no off-target cleavage, no chance of insertional mutagenesis is also here. So there are several ways to reduce this insertional mutagenesis. You create a self-inactivating region by removing the enhancer and promoter regions, or you introduce some specific zinc finger nucleases. Now, which one is better, adeno or retro? Adenovirus is a DNA virus. So it remains as an episomal DNA without integration. So no risk of any insertional mutagenesis. So what is the conclusion? Which one should be better for us? Let us go further. We will get the answer very soon. Adenovirus mediates its gene transfer in this way. That is very simple technique. This is the viral DNA. This is the new gene, that is the red colored part. This is the new gene inserted here. This area is the new gene inserted here. And this viral molecule carries is packed into the adenovirus fudge or the viruses. These adenoviruses then enter into the membrane and then into the nucleus and inject simply the therapeutic DNA into the nucleus like this. Our previous questions will be answered depending on the purposes these adenoviruses and retroviruses they serve. Adenovirus vectors are very useful for tumor oncolysis and vaccination purpose. Now, adenovirus, you know that they can be made tumor specific, I have already told, by certain genomic components, by altering its certain genomic components or adding glycoproteins or specific antibodies to it. Genetic modifications can be carried up to the level of specific tumor killing factors like TNF alpha. It can be incorporated into it. So it will give a strong immunogenicity against the tumor cells. So two immunogenicities will be added together. One immunogenicity of the adenovirus itself and another immunogenicity of the tumor necrosis alpha that has been introduced into the therapeutic gene. And not only that, the adenoviruses, they are also capable of expressing some extra immunogenic genes like gamma interferon, granulocyte monocyte colony stimulating factors, and so many other immunological genes. So it can introduce a battery of immunological genes into the tumors, which can express themselves into immunological protein molecules into the tumor cells that can kill the tumors very easily. The second one, as we all know now, is the use of adenovirus as vaccination purpose. It is a very good candidate vector for generating immunogenic response against various viral proteins, particularly of pandemic in nature, like viruses causing influenza, HIV, and SARS-CoV-2. So if we keep this gene therapy, then you can call this DNA vaccine that has been developed through this uh, adenovirus vector as also a very miniature type of gene therapy. Because here also, you're not modifying the genes of our body directly, but you are introducing some external genes in our body which are enhancing our immunological productions. What is done against the coronavirus? Covishield, for example, is a DNA containing adenovirus, a vector containing DNA vaccine. What is that done here? The coronavirus spike protein gene is introduced into the replication deficient adenoviruses. So this is the spike protein gene, and it has been introduced into the replication deficient adenovirus molecules. Then you grow the virus molecules into specialized cells, purify the recombination gene receiving viruses, formulate the adjuvant molecules for the virus, for the vaccine, and inject into the patient. The adenovirus will enter into the cell very easily because they are masters to attack the cells, particularly human cells. They will enter the muscular cells and the viral spike proteins will be produced. Now your body thinks that there is an infection due to the coronavirus spike protein. So it will generate immune response against the spike proteins in form of antibodies. And you know that whenever antibodies are produced, they keep some helper T cells, which are gradually converted into memory helper T cells. And whenever there is a second antigen response, for example, by a booster dose or by a true coronavirus spike protein, these helper cells will identify that, yes, I have been exposed to this already before, and will produce huge number of antibodies, which will protect you against the disease. So DNA vaccines, they are also a type of gene therapy. In fact, we are having that in our body as a regular phenomenon in these days. Whenever we are taking the Covishield virus vaccines, that is the adenoviral DNA vaccine, or whatever Sputnik or the other DNA vaccines, you know that you are undergoing the small type of gene therapy always. Now, what are the limitations? Because limitations, if you do not mention the limitations, your success is not complete. So limitations of the adenoviruses are also there. If not designed properly, by removing its replicating gene, it can be inserted non-specifically into non-tumor cells, and a huge loss of normal cells may result. This is very easy to understand. And this most important one, or the rather more important thing, is that we have been exposed to this adenovirus several times since our childhood, because it is a very common virus causing common cold. And so a pre-existing immunity in the host cells may kill it before it reaches its target cell. So what happens if the adenovirus here, this molecule is killed here in between its way before reaching the cell? So this problem has to be overcome. Otherwise, how you can successfully give the coronavirus dose 1 and dose 2? Now, this problem can be overcome by using different strains of the virus which generally do not infect the human beings. Say, for example, the human adenovirus 26 against the common human adenovirus 5 number 5 strain. Or you can use a different strain from a different primate, like adenovirus 3 from the chimpanzee, that is C adenovirus 3 strain, against which our body has no immunity or any pre-existing antibody. Now the second question will be derived that whenever you give the first dose with this CAD3 strain, your body will be exposed with this adenovirus strain and will start producing helper T cells against it. So what will happen when you give the second dose after 90 days? Will the pre-existing immunity not kill the adenovirus itself then before it reaches the target? Here the answer is that you can use a different strain of this second booster dose for this particular adenovirus strain. So for example, if this is for the first booster dose, it is C83, then for the second booster dose, it will be, say for example, C8012 or 11. So in this way, more immunological tolerance against the adenovirus can be achieved so that the vaccination program can be made more successful. The second one is the adeno-associated virus. This is also a type of DNA virus, but it is a rather basically it is a single stranded DNA virus, and it needs a little bit of help of this your adenovirus gene to be replicated properly. Say for example, this adeno-associated virus being a single stranded DNA, if it contains a therapeutic gene here, then it must combine with the adenovirus genome also before it can package itself in a competent vector that can carry the transgene. So here you can see these are the genes of your adenovirus assisted adenovirus, that is this one, the capsid protein. This also belongs to this assisted adenovirus or associated adenovirus. But these two definitely need the help of this adenovirus gene. And that's why it's named as assisted adenovirus. The advantage of this vector is that it has got a very strong capsid protein, and you can alter the specificity of this vector very easily by altering the amino acid sequence of this capsid protein. You don't need to put any extra glycoprotein receptors or antibody receptors here. Just by altering the amino acid sequence of this capsid protein, you can also increase its target specificity. The first gene therapy product that was marketed in the European countries, that was against the severe lipoprotein lipase deficiency. It was one of the major milestones in the gene therapy in 1912, that when a gene therapy product was marketed officially in the European countries that could treat the lipoprotein lipase deficiency. And this was made with the help of this recombinant assisted adenovirus vectors. Do you know what was the cost of this drug then? That is Glybera. It was fabulously costly. One treatment regimen to a patient, it costed about your $1.2 million. $1.2 million. So due to this huge cost, that means this cost was put by the company, definitely who produced this novel drug. But due to this huge cost, European countries could not continue it for a long time. And the American countries, that is the United States, they didn't start it at all. And now this drug is no more available in the European countries, as well as it was never available in the United States. So some things matter even after the success. The third important one, DNA virus, is, that is the herpes simplex virus. This is particularly a natural neurotropic virus, and hence its importance is mainly with the neurological disorders like sensory illnesses like your chronic pain. But it can be very effectively used also against the oncolytic therapies, say for example melanoma, because they can produce high amount of GM-CSF, that is granulocyte macrophage colony stimulating factor, and that will be used to produce immunological cells, particularly the T cells within the melanoma or the cancer cells. Now, ex vivo gene therapy that I have already, till now we were discussed the vector based, that means the in vivo gene therapy. Now, if we come to this ex vivo gene therapy, we have already discussed it previously, so I'm not going into it. You can see the figure for a while. You can put your questions if you have any. I think that you have many questions, but I will rather like to show you a particular typical model for ex vivo cancer gene therapy. What is done here? You first construct a therapeutic target gene against, for example, any cancer, say for melanoma or lung carcinoma, etc. Then what do? You insert the gene into a particular virus or particular plasmid, and then you modify those particular virus in the cultures. That means the virus infected cells into the culture. Before that, you take the T cells from the patient and you insert your therapeutic gene, say for example, GM-CSF producing gene into the T cell with the help of a virus like adenovirus or AAV or whatever you like. Herpes simplex virus, you insert it into the cell, you select the cell and expand those cells which have received this recombinant DNA, and then reintroduce these engineered T cells into the body, where they are going to produce huge amount of immunity. So that is going to kill the cancer cells. Advantages of this ex vivo gene therapy are that human body is directly exposed to the therapeutic genes without being exposed to external vectors. That means you need not enable or you need not make the human beings compelled to receive the external viruses inside their body. Now, that is why, as the cells are from the same person, so least chance of immunological or graft rejections like that. And furthermore, in ex vivo process, these type of genetically modified cells, they can be selected and amplified and expanded further before reinfusion can occur. So definitely they are going to increase the effectivity of the gene therapy. I will discuss it later. This is not to be discussed here, in fact. This stem cell induced thing that will be discussed in your next classes, I think. Rather, I will like to discuss this particular technique of gene therapy which brought the Nobel Prize in 2020. That is the CRISPR technology. That means that it's clustered regularly interspersed short palindromic repeats and Cas enzyme, that is CRISPR associated enzyme, which induce cuts at specific areas of the gene by the designed endonuclease molecule. What they do, this is the CRISPR activated or CRISPR specific enzyme, say for example Cas9 enzyme, and this Cas9 enzyme, when it is guided by a particular guide RNA into the target area of a particular mRNA, it will precisely cleave the target mRNA. And now if you introduce a therapeutic gene at the cleavage point, this therapeutic gene can be very easily and handily inserted into the gap. So CRISPRs are natural scissors which cut the mRNA at some target points, and you can dictate the cutting or excision of your target by creating different types of CRISPR activated or CRISPR specific Cas9 enzymes, and you're changing the sequences of the guide RNA. Okay, this is important because this CRISPR targeted enzyme system has already been targeted for several diseases, say for herpes simplex virus keratitis, Leber's congenital amaurosis, transthyretin amyloidosis, cervical cancer, that is a very common cancer killing millions and millions of people throughout the world, and also ex vivo cell-based therapies. And the Nobel Prize of the 2020 was particularly an all-female Nobel Prize in the field of chemistry that proved the capability of the female scientists in the field of chemistry. They were Jennifer Doudna and Emmanuel Carpentier, who received the Nobel Prize in 2020 for discovering this CRISPR-Cas system has got a very potential capability for treating so many gene transfers, gene therapies. So these are the different types of diseases which can be treated by the gene therapies, starting from the cystic fibrosis to neurofibromatosis, etc. Huntington's disease, myotonic dystrophy, so many diseases are there, you will get them in books. They may be autosomal recessive, autosomal dominant. This only reiterates the importance of the gene therapy. This is the process by which you can treat hemophilia using gene therapy. And now coming to the last topic, that is the most interesting one, the germline gene therapy. Till now we were studying about the somatic line gene therapy. That means the gene therapy that we have been studying from yesterday to today, they only limited their effect to the lifetime, maximally to the lifetime of a single person. It is not carried to the next generation, definitely. But in the germline gene therapy, you are modifying the germ cells. Say, for example, you have got an inherent mitochondrial disorder, and you know mitochondrial disorders are inherited maternally. So, and you do not know that mitochondria is present in how many numbers in which cells. So it is very difficult to treat mitochondrial disorders by somatic cell gene therapy. So germline gene therapy has been approached for it. What is done? From the defective patient couple, the nucleus, that is the healthy nucleus, is removed. Because nucleus is healthy, DNA may be defective. Nucleus healthy, nucleus is removed. And from the healthy donor couple, the healthy mitochondria, they remain and the nucleus is removed. Now this healthy nucleus from the defective mitochondrial couple is introduced into the cell containing the healthy mitochondria of the donor couple. And then they are developed into the test tube to 6 to 8 day old embryo that is free of mitochondrial disease, and then it undergoes in vitro fertilization or in vitro implantation, and it develops into a baby which is free from this mitochondrial disease not only, but his next generation will be also free from all mitochondrial disorders. So it is a permanent solution. Permanent solution. So should we convert this germline gene therapy completely? We exchange this germline therapy completely for this somatic cell therapy? What are the yes and no's? Potential benefits of germline gene therapy, do they outweigh the potential risks? For them, in favor of the germline cell therapy are that germline gene therapy enables the correction of disease-causing mutations that are certain to be passed on from generation to generation. So this is the only way to treat the diseases which pass from generation to generation. And also it spares the future generations from suffering from the disease. But what are the disadvantages? The effects of gene therapy are too unpredictable. We have learned it from yesterday. Even if the therapy successfully cures the disease, other mutations, say for example, insertional mutations can be there, and then they will be propagated from generation to generation also. So you are going to create, by a small mistake, a generation of human beings who will carry the induced insertional mutations for a long, long time. Does it consider the individual's human rights? For this point of germline therapy is that no unborn child, whether conceived naturally or through IVF or through germline gene therapy, is able to choose their genetics. So what is the harm in using this germline therapy? But against the point is that individuals produced through germline gene therapy cannot give their consent. So if they suffer by some insertional mutagenesis, they will curse that the thing occurred without my consent when I was in utero. Is it ethical? It depends on several factors. But basically, it depends on the state of the society and the state of the disease. Is it affordable? Definitely it is affordable to rich countries who will produce better generation, disease-free generation, and definitely it is not affordable and they will be left with a defective generation or disease generation, creating a severe disparity throughout the world. Some countries, some affluent countries will have completely generations free of all diseases, and some countries will be there in this same world who will have generations suffering from the same diseases. So lastly, if this germline gene therapy really needed or not, it is a $1,000,000 questions. For this, with the widespread use of germline gene therapy, particular genetic conditions could be eliminated from the population altogether. This is a boon to the population, no doubt about this. Say, for example, treating the Huntington disease and some other disease, which can be eradicated just like we have eradicated the polio and measles from our human history, from the human civilization. But against the point is that germline therapy would never be effective enough to rule out the need for post-fertilization screening of the embryos. So whether it is successful or not, for knowing that, you have to do this post-fertilization screening of also. So if you have to go for post-fertilization screening of embryos, then what is the use of making this germline therapy? Because in the post-fertilization screening, you can find it out and you can abort the child if it is not okay. So that question lies before us now. Should we go for sowing only the healthy seeds in this world? Like only healthy seeds must be sown. This was a very popular slogan in 1939 to 1945 during the Nazi regime, when Hitler carried out his experiments with the eugenics. That means to create pure Aryan race in Germany. So he made this slogan, only healthy seed must be sown. Generally, this became a very popular slogan then. Or we should be ready for this. Convert bad news to good news. We welcome the diseases, we understand them, and we treat them to their therapeutic success. So this is the mainstay of the gene therapy that has been discussed since last 2 days. We have discussed many things, and I think that they might have been a little more for you. But gradually, if you see them later, it will be more beneficial to you if you have some books, good books on gene therapy or good journals on gene therapy with you, and then you see the slides, then you go through the lecture. Whenever you need to see some clarifications, you go through the books, you go through the nets, and it will be clear to you, much more beneficial to you. So thank you very much for hearing. I finish my lecture here.
Thank you very much, sir, for your wonderful presentation. It was very much informative. Still, it was very much interesting for all the students, I think, and as far as the faculty. So in our chat box, we have received one question.
So should I take the question from the chat box?
Yes, sir. I will just read it out for you.
Okay, thank you
That
so
what
much.
is the yes, sir. What is the relevance of gene therapy in treatment of diabetes mellitus in current Indian scenario?
Okay, the thing is that diabetes mellitus is a multigenetic factor. In fact, so many genes are involved in diabetes mellitus. So it is a very difficult task for achieving a cure for diabetes mellitus because I've already mentioned that gene therapy is mainly successful for those diseases which are mainly monogenic in nature. So if you want to treat diabetes mellitus, then you have to consider the type 1, type 2, then this MODY separately, then pregnancy-induced or hyper-induced diabetes, gestational diabetes mellitus, and in each of them you find different genes are working. So definitely it is a tough job, but it can be done easily or not easily, relatively more easily with the help of the recent techniques like microRNAs, which can target many genes together. So much more larger amount of researches with these microRNAs are required because you need a multi-genetic approach to treat this disorder with the help of gene therapy.
Thank you, sir. And
Any
no more
question?
questions, but
No
many
more
of the
question. Why?
many of the attendees, sir, no, sir. Many of the attendees have asked for the book and the slide references.
Yes, I couldn't hear you, Shantasmita. Please repeat.
Okay, sir. Many of the attendees, yes, sir. Many
Yes.
of the attendees have asked for the references. That is from which book or from which materials they can get enough information about this gene
Okay,
therapy.
okay. I will give them. I will give them. So when I should give them? Can I post the references? I think that I can post the references uh, as a file, as a Word file in the SCCLMP site,
Okay, sir. That
or
will be beneficial for
that,
them.
or how can that be transmitted to the students also? Okay, we will discuss it later, and
Okay, sir.
we will definitely supply you with the references. Definitely, I have got a battery of references because within these 5 to 6 days, last 5 to 6 days, I have gathered so many research papers, so many journal that they will be very valuable things for you.
Yes, sir.
Okay, so any more question, students or anyone?
No, one of the attendees have asked for any gene therapy for ovarian cancer. Is there any gene therapy for ovarian cancer?
There must be. There must be the particular type of gene therapy for ovarian cancer. But during the search of my literature, I got most of them for small cell lung cancers, then like Duchenne's muscular dystrophy, cystic fibrosis, myotonic dystrophy, then macular degeneration, then cardiac disorders, lipid storage disorders, lipid profile disorders like that. But I didn't get a chance to look into their role in ovarian cancer. Definitely, I will look for that, and I will give it whenever I get a definite source, a reliable, definite source for a gene therapy in ovarian cancer. I will supply it. Okay.
Okay, sir. No more questions for now.
Okay. So thank you so much. Thank you, SSLMP, for arranging this lecture or this webinar. Thank you.
Yes, sir. Thank you very much. Thank you,
Okay.
sir.
Yeah.
Thank you to all the attendees also.